Assalamu alaikum. I welcome all in today's lecture on pathology. I am Professor Shamsudjaman, Professor of Pathology. This is my 7th lecture. In the 6th lecture, I have told you about the fatty liver. Today, irreversible cell injury. Now come to irreversible cell injury. It is that type of cell injury from which the cell cannot come back to normal state after removal the damaging stimulus. So, type of cell injury from which the cell cannot come back to normal state, after the removal of damaging stimulus. So we can recapitulate. If the cell can come back to normal state after removal of damaging stimulus, it is called reversible injury, and reverse is true. If it cannot come back to normal state, this is called irreversible cell injury. It is also called cell death. So, again we can recall, in case of reversible cell injury there is point of return, but in case of irreversible cell injury there is no point of return. Here no point of return. So this is irreversible cell injury. Now come to what are the characteristics of irreversible cell injury. Characteristics of irreversible cell injury. It means what are the characteristics of the cell when it undergoes irreversible cell injury. The first and foremost characteristics: cell membrane damage or disruption. So, cell membrane disruption. Then lysosomal swelling. You know, lysosomal bag contains enzymes. These are called lysosomal enzymes. That bag undergoes swelling. Later on, what happens? You know, mitochondria is the powerhouse of the cell. Within the powerhouse, there is severe vacuolization. If there is severe vacuolization within the mitochondria become dysfunctioning. And if the mitochondria become dysfunctioning, cell undergo death. Another one, there is amorphous densities in the mitochondrial matrix. Amorphous densities in mitochondrial matrix. So, if there is cell membrane damage, lysosomal bag swelling, and there is dysfunction of the mitochondria, definitely cell undergo irreversible injury. This is nothing but cell death. Now come to what are the biochemical mechanisms of cell membrane damage? So this is the cell, this is the nucleus, the membrane undergo damage. Now go to biochemical mechanisms. Biochemical mechanisms of cell membrane damage. What are the mechanisms? The first and foremost mechanism is lipid peroxidation. Lipid peroxidation. We know cell membrane is lipid bilayer. It is composed of lipid and also protein. If lipid peroxidation, what happens there? There is activation of phospholipase enzyme within the cell membrane, and this phospholipase causes breakdown of the lipid of the cell membrane, and this is lipid peroxidation. The phospholipids of the cell membrane is destroyed or lost, so the cell membrane become damaged. This is lipid peroxidation. Another one is cytoskeleton. Cytoskeleton abnormalities. Cytoskeleton abnormalities. You know, microfibules, microfilaments, all are these cytoskeletons. It is associated with the strength of the integrity of the cell membrane. If cytoskeletal abnormalities occurs in irreversible cell injury, the cell membrane undergo damage. Damage by lipid breakdown products. You know, during lipid peroxidation, lipid undergo breakdown, and during lipid breakdown, there are certain products are released. That products are very much injurious for this membrane. So damage of cell membrane by lipid breakdown products. Loss of intracellular amino acids. It is also associated with lipid cell membrane damage. So this is the biochemical mechanism of cell membrane damage. There is also another mechanism that is associated with cell membrane damage, oxygen derived free radical injury of cell membrane. So this is biochemical mechanism of cell membrane damage. So this is all about the irreversible cell injury. Now come to what the types of irreversible cell injury. That means what the types of cell death. Now go to types of irreversible cell injury or types of cell death. One is necrosis. And another is apoptosis. Today I shall discuss about the necrosis. Now come to necrosis. Before going to discuss about necrosis, we can recapitulate. Suppose this is tissue. It is composed of innumerous number of cells. All the cells are functioning, active anatomically and both physiologically, functionally. These are functioning cells. This is a tissue. All cells are functioning. If within the living tissue, this is living tissue. If due to any causes, what may be the cause? whatever may be the cause, if one or more cells undergo death within the living tissue. Tissue is very much living, but a few number of cells undergo death here. After the death of the cells within the living tissue, these cells are dead cells within the living tissue. So there are some changes develops within the dead cell in the living tissue. All the changes that occurs in the dead cell in living tissue, the summation of the changes is called necrosis. Again we can say some changes occur in the dead cells. The dead cells are within the living tissue. The changes that occurs within the dead cell collectively known as necrosis. So we can define necrosis, sum of morphologic changes. Sum of morphologic changes. Sum of morphologic changes following cell death. Sum of morphologic changes following cell death. Following cell death in the living tissue. Following cell death in living tissue. In living tissue is called necrosis. So the changes that occur in the dead cells within the living tissue is called necrosis, and this is due to this. Why these changes occurs? These changes are due to progressive and degradative action of the enzymes of the dead cells. The enzymes present within the cell nucleus, within the cytoplasm, and also in the lysosomal bags. So it is due to progressive degradative actions of enzymes. The enzymes present within the cytoplasm of dead cell and within the lysosomal bags are associated with the changes, and all changes collectively known as necrosis. So this is the definition of necrosis. Now come to, if a cell undergo necrosis, and this is the nucleus, what are the changes that occur within the necrotic cell? Summation of morphologic changes. What are the morphologic changes? Changes in necrosis. Changes in necrosis. So change may occur in the cytoplasm and change may occur also in the nucleus. First, changes in cytoplasm. Changes in cytoplasm first. Changes in cytoplasm. The first and foremost change in the cytoplasm is loss of basophilia. You know, the cytoplasm of normal cell, some portion is basophilic, some portion is eosinophilic, and basophilia is due to RNA. In necrosis, RNA is depleted, loss of RNA, so there is loss of basophilia. The loss of basophilia, it is due to loss of cytoplasmic RNA, because RNA is associated with basophilia. In necrosis, the RNA undergo destruction. Another change, loss of basophilia, increased eosinophilia. Increased eosinophilia. In necrosis, cytoplasmic amino acids undergo loss, denaturation. Then cytoplasmic proteins, cytoplasmic amino acids undergo denaturation, And denatured protein or denatured amino acid, when it is stained by hematoxylin and eosin stain, it takes eosin color. So due to denaturation of protein cytoplasmic, the cytoplasm become eosinophilic. It is due to denaturation of cytosolic proteins. Then homogeneous cytoplasm. Normal cytoplasm is not homogeneous, rather it is a bit heterogeneous because it contains glycogen and many other things. The loss of glycogen of the cytoplasm in necrosis makes the cytoplasm homogeneous. It is due to loss of glycogen. Then vacuolization in cytoplasm. Cytoplasm will show extensive vacuolization in necrosis, later on moth-eaten appearance. We can cite an example. If we want to see a diary or a book, it was written by my forefather about 70 years ago or 80 years ago. If you open the diary, we will see each and every page of the diary is porous. It is due to bite by insects. The cytoplasm of the necrotic cell will show some porosities or porous. It will look porous. This is due to eaten by moth. Actually, it is not eaten by moth. It looks like moth-eaten appearance. So there is a cytoplasmic changes. Now come to nuclear changes or changes in nucleus. Changes in nucleus. What changes occur? You know, this is cell, this is nucleus, this is chromatin or chromosomes. In necrosis, within the cell, there is first condensation of the chromatin. If the chromatin under condensation, the nucleus become smaller in size. And if they become smaller size and condensation of chromatin, it becomes dark colored. So first is pycnosis. Pycnosis of nucleus. What it means? It means condensation of chromatin with shrinkage of nuclear size and darkness of the nucleus. It is called pycnosis. What is this? The condensation of chromatin. As there is condensation of chromatin, the size of nucleus becomes smaller. The shrinkage of nucleus. So there is shrinkage of nucleus. So shrinkage of nucleus. As this nucleus is smaller in size, pycnotic in size, the color become dark. So shrinkage of nucleus and darkness of nucleus. This is called pycnosis. The second change in the nucleus, within the pycnotic nucleus, the chromosome or chromatin undergo lysis. This is called karyolysis. The second change is karyolysis. Breakdown or lysis of the chromatin is called karyolysis. How it is undergo lysis? It is due to DNase enzyme. Lysis of chromatin. Lysis of chromatin by DNase enzyme. The last change, breakdown of the pycnotic nucleus. It is called karyorrhexis. What it means? Breakdown of pycnotic nucleus. So these are the nuclear changes and cytoplasmic changes in necrosis. We are talking about irreversible cell injury. It is two types. One is necrosis, another is apoptosis. We are talking about the necrosis. Now come to types of necrosis. Types of necrosis. Now come to types of necrosis. Basic two types. Basic two types. One is coagulation necrosis. Coagulation necrosis. Another is liquefaction necrosis. So one is coagulation necrosis, another is liquefaction necrosis. These are the basic types. Other than basic types, there are other special types. Other special types. What are other special types? Now come to other special types. Now come to other special types. One is fat necrosis. Another is caseous necrosis. Another is gammatous necrosis. Another is gangrenous necrosis. Another is fibrinoid necrosis. Fibrinoid necrosis. So this is the types of necrosis or classification of necrosis. Dear audience, respected audience, today up to this, thanks all.